name is Jian JIN, a PhD student from GEPA lab, Nantes University, France. Today my topic is biomass and hydrocarbon production of microalgae Botryococcus in continuous culture. I will present in these 4 parts. The first is the background and the review. From this figure, we can see the world total petroleum consumption increase every year. To find a sustainable production of biofuel is meaningful. The third generation biofuel is using microalgae to produce biofuel. It's more efficient and no competition with food. The microalgae biofuel is not only biodiesel but also biohydrocarbons. Biohydrocarbons is the main component of fossil fuel. It has high caloric value and compatible with existing combustion engine. The question is, how can we find a bioresource to produce biohydrocarbon? From the first table, we can see most of organisms can produce hydrocarbon, but the contents are very low, generally less than 1%. From the second table, we can see there are some organisms are hyperproducer. They can produce high content hydrocarbon. And Botryococcus is a microalgae. It can use CO2 to produce hydrocarbon, so it's more eco-friendly. Here is the biological model of Botryococcus. The cells of Botryococcus are aggregated together to form colonies, and hydrocarbon are accumulated in the cell in the lipid bodies. Also, hydrocarbons are accumulated out of the cell in the extracellular matrix. There are different types of hydrocarbons. All these kind of hydrocarbons can be used as energy biofuels. This microalgae is potential but understudied cell factories. For example, the relationship of hydrocarbon production and nitrogen conditions, environmental pH are not sure yet. pH are very important for the organisms. It can affect enzymatic activities, inorganic carbon speciation, and macromolecules transportation. However, from previous studies of pH effect on Botryococcus, only in animal and without continuous pH control. In recent years, there are more than 10 studies of Botryococcus in photobioreactors. But among these studies, only 2 studies control the pH, and they control the pH at 7.5 or 7.2. However, we don't know this 2 pH is the optimal pH for this microalgae to produce hydrocarbon. So from now, there are few reports of pH effects on Botryococcus in continuous culture. So in our study, we try to do this kind of study in continuous culture. Continuous culture can we can get more reproducible and reliable result. And using successive steady state, we can get biomass productivity and hydrocarbon productivity as a function of environmental factors. Here is our continuous culture setup. In the center is the photobioreactor. Here the microalgae were coated in, and the left is the pH control system. The right is the feed medium and harvest bottle. For our experiment, we set all other factors as constant, only change the pH step by step from 5.5 to 8. Next, let's see our result. Here are two figures. The first one is biomass productivity and hydrocarbon productivity. From this figure, we can see biomass productivity are relatively unaffected by pH. However, hydrocarbon productivity seems significantly affected by pH. And not only the productivity, but also the hydrocarbon content also significantly affected by pH. Surprisingly, from pH 7.5 to 6.5, the productivity increased significantly. Then we wonder if this kind of change is only caused by pH or maybe other factors has an effect on this. For example, the nitrogen condition. From other studies, nitrogen conditions are very important factor for the microalgae. Then we did nitrate balance analysis. By using ion chromatography, we can measure the nitrogen condition. When we're using the AF6 medium as a feeding medium, from the outside, we can't detect nitrate. So our culture in nitrogen limitation conditions. Then we try to use nitrogen-rich medium. Then from the outlet, we can detect nitrate. So in the second conditions is nitrogen no limitation conditions. For the following experiment, we use these two conditions and choose pH 6.5 and 7.5. Here is our result. The left is the biomass and the right is hydrocarbon. For the biomass, we can see both on the nitrogen no limitation and the limitation conditions at the pH 6.5 and 7.5. There is no significant difference. However, for the hydrocarbon, whatever the conditions, pH at 6.5 are significantly higher than 7.5. We also did microscopy and fluorescence observation. Here, by doing now-rad fluorescence studying, we can the yellow part is the hydrocarbon. At the 6.5, it seems more hydrocarbon accumulated in the out of the cell in the extracellular matrix. However, at 7.5, more hydrocarbon accumulated in the cell. So maybe the hydrocarbon excretion process also pH dependent. Last is the conclusion and the perspectives. Here from this experiment, we confirmed that continuous culture is an efficient method to investigate effects of factors on hydrocarbon productivities. From our result, we discovered that nitrogen no limitation conditions could increase biomass and hydrocarbon productivities. And from our result, we found that pH 6 to 7 could be the optimal pH range for hydrocarbon productivities in Botryococcus cultures. This could be the pH-dependent excretion process, or pH can affect the enzymatical activities, or could be strain or race dependent. Further studies is undergoing. Thank you. Any questions?
You say that it could be pH dependent excretion of the this one. Did you check the pH intracellular?
we didn't check. We only control the environmental pH, and then at this pH 6.5 and 7.5 conditions, we sample and observation. Actually, we are not sure for this conclusion. It seems at pH 6.5, more hydrocarbon are accumulated out of the cell, because from the literature for this microalgae, hydrocarbon first biosynthesis in the cell, in the lipid bodies, then it can excrete out of the cell to the cell surface, then accumulated out of the cell. So we guess maybe the difference could be caused by pH. pH could have an effect on this kind of excretion process, but we didn't check the intracellular pH.
Indeed, if you check the intracellular pH, it would be much more easy to know.
Good suggestions. Thank you.
More questions? Okay, then thank you.
Okay, thank you.